recording. Okay. Start to share. All right, so my apologies to those of you that are starting to watch the video. I forgot to turn it on, but we're partway through doing the review of characteristics of polynomials. So we just established on this graph that there are 1, 2, 3 x-intercepts. Oh, I'm gonna do it this way. So I'm gonna put 3 here. Use my pencil. This is graph number 2, yes. And then I need to write the coordinates of them. Now, careful. Oops. So the coordinate of this one is -5 and 0. I am very, very fussy. If you're going to write me an intercept, it needs to be written as a point. Now this one, I don't know, -2.5 and 0 and 0 and 0. All right, go to the next picture. All right, I think you guys can do this on your own. Let's make this up here. There we go. I think you can, I would mark it on my graph, and then I would write out my coordinates. All right, Clarissa, how many x-intercepts are there on this graph?
2.
This is my x-axis.
1.
What? And it's right there. And where's my sheet? I reprinted my book because I made such a mess of it. If not now, if you guys get lost at some point, there is this page, page 6, and it tells you a lot of this stuff, and you can double check. So that's a linear equation, and it says it should have 1 x-intercept. That's all we're allowed. So you can catch yourself and double check. Anyway, Clarissa, can you tell me the coordinates of this point?
-1.
And?
3.
No, 0.
Oh.
I just need this one. When you do an x or y coordinate, the other one is always a 0. So my brain, actually, I never have, how do I explain this? I don't think of x-intercepts. I think of my phrase as x, intercept y is 0. It's all together as 1. Okay, I'm going to tell you guys a stupid story. I should turn off the recording for this, but you know, everybody needs to know this. So when I was in university, I was chasing a guy and his name was Eric. It didn't go well, but he was my heartthrob. He was just my dream boy. And I really, really had a crush on him. Didn't go anywhere. Fast forward, I'm having my child. My child's name is Eric. Named after, yes, see, you get it. Named after my husband's uncle, who I've met, and he's a really nice guy. But for the first year of my son's life, every time I went home or met one of my high school or university friends, and I said, this is my son, Eric, they'd all give me this stupid look. So I started to say, this is my son, Eric, not named after the guy from university. And it just, that was his name for the first little while of his life. So your brain needs to say x-intercept, y is 0. It should just flow as 1. X-intercept y is 0. Y-intercept x is 0. The other one's always 0. Cool. There you go. Now you got way more information than you need to know about me. All right, let's look at number 4. All right. Jose, you are just joining now. This is the assignment we're working on. It is a big sheet, 11 by 14, so double size. But the most important thing is make sure you find the one that says practice here so that you're getting the answers in the right spot. And we are looking for x-intercepts. I'm going to do one more together with you, and then I will let you guys do some, and we'll correct them together. All right. Excellent. Just a heads up. Anything you chat to me is showing up on the big screen in here. So just keep chatting, it's fine, but just a heads up. All right, Jose, if you look at this, we have the y-axis and we have the x-axis. So the x-axis is always a horizontal one. And this is the graph that we care about. How many times does this curve cross this blue line?
Twice.
Excellent. Yeah, there and there. So on my chart, I'm going to write that it crosses twice. And then I'm going to do my coordinates. Now this one, I'm going to guess because it's not exactly on the line. So if you put 0.3, 0.4, 0.5, cool, that's fine. For this one though, because it crosses right at a corner, I would expect you to write 3 and 0. So I would like you all to continue. I'm going to stop for a minute, and I want you to go through the rest of the graphs. And for each of them, I want you to write the number of x-intercepts and the coordinates of them. While you're doing that, I can check what's going on with everybody here. To me, it's silly how much better it is once you highlight that. I get so confused. And I know that this one picture is messed up. On mine, I don't know, it shifted on yours, so it's a little harder to read, but keep going. You're good. I'll stop bugging you.
It's hard for the y-axis of that one.
Oh.
I can't see where it is.
Oh, then never mind.
I mean, you
No, you can't. No, don't worry. In case anybody thought I wasn't actually here, I'm waiting to do that.
sometimes speeding
How about those of you who are online? Give me a thumbs up when you're done. Or chat me if you're stuck. I'm so glad you're Zooming this morning because this stuff, it looks scary. But once you start doing the actual analysis, it's not so bad.
Yeah. Is this all it is?
Yeah, pretty much. The flip side of this has got a bunch of equations, but if you plug those into Desmos, you'll have the picture, and then you're just doing this again. All right, Driana, where are we on? Number 5, what are your answers? And none, exactly, right? Here's my x-axis. My graph doesn't touch it. Cool, not a problem. All right, Clarissa, number 6. How many times does it cross the x-axis?
Twice.
Beautiful. There's one. There's one. Kate, what are those coordinates?
-1 and 2.
But I know you wrote them as points like that. Please. If not, fix it now.
I didn't do that.
Okay. I'm fussy. I want it done like that. The worst thing you can do is -1 and 2, which would be -1, which would be like a point up there. I'd give you half marks, but why not get full marks? All right, number 7. Jose, are you there?
Yep.
Okay. How many times does my line cross the blue one?
Is that the middle?
This one. So this is the line I'm caring about. How many times does it cross just this blue line?
6 times?
It crosses once.
Once.
Right there. So that's where they intersect. So that's going to give me 1 there. And my coordinates are, it's 4 over. That's where it crosses. And then we put the 0 to show it's an actual point. It's none up. All right, Sammy, last one.
Number 8.
Yes, please.
1 and 0. And it crosses once.
Okay, so it crossed once. It's right there. And it is 1 and 0. Ta-da! You now know all about the x-intercepts. All right. Oh, you know what? Why don't you do the y-intercepts? We'll do the first one together. So the y-axis. Cleveland, which one of the axes is the y-axis?
The vertical one?
There you go. I know you knew it. You just lost the word vertical, didn't you?
Yeah, I get them mixed up.
That's okay. As long as you know that it's the up and down one, we're perfect. All right, come back. How many times does it cross the y-axis?
Once.
Where? Give me the
At 3.
coordinates. Okay, give me the coordinates.
3. I don't know. Is it +3?
Yep.
Okay.
Oh, you can't see it, but the x-axis is down there. So the coordinates here are 0 and 3. Y-intercept, x equals 0. So watch when you're doing these. If it's the x-intercept, the number is where the x is. If it's the y-intercept, the number is where the y is. I believe you all have the skills to do the rest of them. Y-axis, y-intercept, all the way across.
Except for the second one.
Oh, yes. Here, this is number 2. Yours on the paper, I don't know what happened. right? This is what you guys have. My graph shifted or something when I printed it. I don't know, but this is what it's supposed to look like. So you now can read your y-intercept. So across the page. And you can see there. But you can do all the way 1 to 8. Okay, no, this one's messed up because if you look at my graph, your graph got caught off at -1. So, yeah, I did that. Okay. You know what, guys? This is my suggestion for this one. Let's just do this. Because afterwards, if you have 0 and 0 for that answer, you're going to look at the graph and go, where did that come from? So get rid of that and just go on to number 3. Don't forget brackets, right? Pumpkins are beautiful. Yep. Got to be crossing one here, yellow line. How's that? That's where your intercepts go. Yeah, actually, they are both of them. That's supposed to be 1 week, 2 weeks. All right, let's do this. So this is graph number 3. And here's my x axis, my y-axis. Cleveland. Oh no, I just picked on you. Clarissa, how many y-intercepts do I have?
1,
Yes. And what are the coordinates of it?
4, and 0.
Almost. Whoops, can't see that. 0 and 4. I just realized I'm making life complicated for us. If it asks for the y-intercept, just put the number where the y is. If it asks for the x-intercept, put the number where the x is. All right. This is graph number 4. Jose. So the y-axis is this one. How many times does my curve cross the y-axis?
1.
Yes. All right. We got once, and it's right there. What are the coordinates of this point?
Zero 0.3.
You got the 0 part right, but it's 1, 2 up. So 0 and 2,
So it's 0.2.
comma 2.
Okay.
There we go. Cool, cool. All right, number 5. Adriana. Whoops. Trying to make this, you can see both parts on the graph at the same time. Nope. If you look on the y-axis, it's the vertical one. There's one of them. It's right there. So 1. And what are the coordinates of it? No! 0 and -4. I'll get you guys yet. So order matters. It's an ordered pair. All of these ones are going to start with 0. You know what you could do? If I was doing this, and I'm doing a whole bunch in a row, instead of having to go, which way does it go? Just fill that part in. And then you're just plugging in the back number all the time. Same thing with this. I put zeros at the back, plug it in. All right, so that was, where are we? That's number 5. And now we are at number 6. Clarissa, your turn. How many y-intercepts?
1.
Yes. And what are the coordinates?
0.1.5.
Yeah. Okay. Just be careful, it's 0, 1.5. But I know that's what you meant, so it's cool. All right. Sammy, what about you? Next one.
1-intercept, and the coordinates are 0, 2.
There we go. There it is right there. 0, 2. Okay, I'm excited with putting the 0 in there to begin with. That's just so much simpler. Just put the zeros in, then do the stuff we have to think. All right, last one. Jose, back to you, my friend. How many times does it cross the y-axis?
Twice.
Nope. Okay, this is my
Or
y-axis.
once.
It's going to be once. It crosses right there. And how far down is that?
So 2.5.
Okay, we need it right there, so it's -2. And there we go. So I'm just going to put this so we can see. Notice the number of y-intercepts is always 1. Always. The number of x-intercepts can change. It can be 0, 1, 2, or 3, depending on the type of graph we have. So this one was a cubic, which is why it's 3. Anyway, 0 and 3, 0, 0, 0 comes first. If it's a y-intercept, and then there's a number where the y is. Sorry, I'm feeling a little pleased with myself. All right, now we're going to do the starting quadrants and the ending quadrants. What I did before I started this morning, not only did I highlight my axes, because I don't know. Every time I look at these ones on Desmos, my brain starts to go all over the place and I can't forget where the axes are. So I highlighted them, and I also marked in 1, 2, 3, 4. I marked my quadrants. I did that all at once, so I only have to think about it once and get it all done. Now, when we're reading a graph, you are reading it from this direction. So you are looking, going this way all the time. So the first thing we can do is, if we're looking for starting quadrants, our choices, it's either going to start in 2 or 3. That's it. It has to come this way. So it's going to start in either 2 or 3. So I'm going to mark that down so I don't screw it up. Those are my only choices. And it's going to end over on the right-hand side. And that's either going to be 1 or 4. Those are the only choices. So look, even if you're completely guessing, your probability of guessing correctly is 0.5 as opposed to 0.25. And maybe the way to think of it, let me try this on for size. Put a star, I'm gonna put a mark here, where my graph starts. The first time you see it. All right, mark, whatever. I went to the dollar store and bought myself highlighters. Again, I'm feeling pleased with myself. Jose, what quadrant is this orange dot in? Is it 1, 2, 3, or 4?
One.
It is over here in quadrant 2. And since we got the graph there, let's do our end. So here's my end over here. Clarissa, what quadrant does it end in? Whoops, can't see that anymore.
1.
Yeah. So this is my start. And just look for the, I guess, the beginning and end of the graph and figure out what quadrants it is. We'll see if this works. All right, put that one away. Now I'm going to look at my next one. And I'm going to put a mark where it starts. And it goes, I don't care what it does in the middle, but it ends down here. All right, Cleveland, what quadrant does this, you can see it, does this one start in?
2.
Yes, I was going to say, wait, but I marked my graph wrong. So 2, where does it end?
4.
Yes. All right, so you should have 2 and 4. Sammy, what quadrant does it start in? The next one. This is number 3. Yep.
3 and 1.
And so 3 over here and ends up in number 1. Cool. Oh, people are going to be so sad if they're not watching this. If not now, later. So mark the ends. Brianna, where are we starting? Quadrant? Yep. Where are we finishing? Nice. Okay, there are 4 left. Finish them up, everybody. And we'll go through them right away. All right, let's give this a shot. So this is graph number 5. And if I'm reading from this direction, Jose, which quadrant does it start in?
Four.
Okay, we got to read this way.
That's 3 and 5 there.
3 and 4. And if you're doing the assignment and you get frustrated and counts down Roman numerals, I don't really care if you renumber them. It's just some weird tradition that we use Roman numerals. I think simply so that you don't get the other numbers mixed up. I'm not going to be too upset. All right, so there we go. Now, next graph. Cleveland, how about you? Where's that starting?
2 and 4.
Beautiful. And that was said, you know, with confidence. Clarissa, how about this one?
2 and 4.
Nice. Sammy, last one. Cool. See, that wasn't quite so painful. All right, so that's what you should have. And now I'm going to teach you all how to cheat. Get out your booklet. Well, I'm not sure I'm teaching you how to cheat. In retrospect, assigning the leading coefficient is a little bit silly, considering you don't have an equation. But we can work backwards, I think. I think so. I don't know, let's try. This is the chart I'm talking about. It's on page 5. Seriously, I do this all the time. I just label them to begin with, and it just makes my life easier. So I want to see, so if we look at this one, and this is a nice linear equation, we are starting in 3. Let me do it the other way. And we end in 1. And that's when I have an odd degree and it's positive. This one, start. And we're starting in 2 and we're ending in 4. I want you guys to do the start and end here. This will work. So for positive even, the start is in 2 and it ends in 1. And for even negative, it's going to start in 3 and end in 4. Now, what does this got to do with anything? Well, yeah, how can I show you both of these at the same time? Ah, what if I put that here? And can we see? All right, move it this way. There, now we can see both at the same time. So this is my chart. Okay, why am I telling you that? You know what that is. So I'm looking down here. It's starting in 2 and ending in 1. And then I'm just going to look on this and find the one that says starting in 2 and ending in 1. And that says my coefficient is positive. So we're matching this to what's on our chart here. And then we're putting in whether it's a plus or a minus. And if we look at graph number 1, it actually looks exactly like that. So we know it's positive. So the next graph says 2 and 4. So if I go through here, it's 2 and 4. And if I look, well, it's got a wobble in the middle, but we still have that same thing. So 2 and 4 is negative. Keep going. No, you're already done. Look at you. Okay. Are you having that moment of it's way too easy? All right, let's see. 3 and 1. Okay, Adriana. 3 and 1, positive or negative? Nice, right there. There's a small part of me that's thinking, how did I teach this before? This is so easy today. Maybe it's just the skill set in here. All right, 2 and 1. Cleveland, tell me about if it starts in 2 and ends in 1, positive or negative?
It's positive.
Right, it's right there. And now we've got, oh, I can bring it up like that. 3 and 4. Jose, from here, tell me about the one that starts in 3 and ends in 4. Is that a plus or a minus?
It's a minus.
Perfect, right there. Sweet. Clarissa, what about the next one? 2, 4.
Minus.
Yes. All right, Sammy. Oh, another one. And Driana. So, did you get it?
I only got one wrong.
Nice.
Too much negative.
All right, sweet. Now, I'm going to show you a new cheat. I could, and teacher-wise, I should, go through yet another explanation of what domain is, but flip your page and look at this chart. This is your favorite chart.
Is that all it is? Really? Like,
Yeah.
for all of them?
Look, people. X is an element of real numbers all the way through. It doesn't matter which one it is. You just write XER. Yep. Just like there's always just one y-intercept, this is always that. That's what you're going to write all the way across. No, you don't have to have the funky. Okay, I would do. You know what? I'm completely okay if you do that. And that. just so you know, I don't lose my paycheck and get in trouble for being super lazy. I'll do it once. What that means, okay, is for any number I pick, get this straight, along this x-axis, wherever I pick, I can go straight up and I'm going to hit the graph. So if I go straight up from here, I'm okay, almost straight. And I'm always, now, when I get here, it looks like I'm going to miss a graph, but in reality, this keeps going. So no matter what, I'm always going to hit the graph. If I start at the x and go either up, or sometimes you got to go down, but you'll always hit the graph. That's what x is an element of real means. Any number that you pick for x, you can plug into the equation and you will be able to get some answer. Range, almost as easy. Let's take a look at the chart. It says, well, this part I don't really care because none of our graphs were constants. But it says anyone that's marked as linear is YER, and anything that's cubic is YER. So let's deal with the, you can do that. I got to put my answers in the top. So quadratic, number 2 is cubic, number 3 is linear, quadratic, oops. We already talked about all those answers up there. So now I'm going to use this, and it says when it's linear, it's YER. So I'm going to take my little chart. This is linear. Y is an element of real numbers. And there's the other linear. Y is an element of real numbers.
You don't have to write that whole thing with little squigglies in there.
It'd be lovely. It would make me happy. Am I going to dock marks? No, because you know what it is. It also says if it's cubic, it's Y is an element of real numbers. So now I'm going to go on my cubic ones. There's a cubic. There's a cubic. So I'm going to go down. And there we go. Look at that. Half of that without a whole lot of work. And what's left are the quadratics. The quadratics require a teeny bit of work. Because, for example, if we look at number 1, when I talked about domain, I said I can draw a line from my x going up or down, and I'm always going to keep hitting my graph. But if I draw a graph this way, so I'm going to cross my y-axis. Do you see that where I'm drawing right now? I'm making a terrible mess, but I'm not going to hit my graph. So any number down here, there for this y of say -1, I'll be nice, I'll draw right on the line. There's no associated x value that we're going to hit. And that's going to keep happening until, well, as I say, I feel really cheap. I should go find a ruler. I hit this point. When I hit there, then I actually have a y value. And this is called the minimum. So all of my y values are going to be bigger than this point. And if we look, it tells you when a is positive, you know what? Do that. Put a smiley face beside it. And put a frown this one. And I always think that's nice and easy to remember. Positive makes you smile. You're going to write y. You know what? We can just do this. Y is greater than or equal to, because I didn't give you much room. So y is greater than or equal to, whoops, and now I need my number. And what I'm looking for is this. And I'm going to say that's about 1.7. So that is what you should have in there. We don't need to, the other stuff's nice, but this, ouch, is good. So y is greater than 1.7. Okay. Now, to explain that, if, right there, we've got a point, and it's 0 and 3, my y value here, that is greater. This number is bigger than 1.7. If I pick another coordinate of this graph, let's say this one, okay, that is about 1.5 and 4. If I'm only caring about the y, this value is bigger than 1.7. And no matter where I get, it's always going to be bigger than this. There's no way, if I look at a y value of like 1, there's no graph there. So that's how we work the range. And now I can skip over graph 2 because it's done. Skip over graph 3 because it's done. And we will look at graph number 4. So the key point you want to find is either the maximum or minimum. And that's where it changes direction. So in this one, it looks like it's right about there. And this is a smiley face. A is positive. A leading coefficient is positive. And if I move this up, yep, we can do that. I'm on number 4. So y is greater than or equal to. So if it's a smiley face, it's great to smile. It's great to be positive. That's how I remember it's greater than sign. And then I go across. And nice straight line, sort of. And that looks like about negative 2.7. On a question like this, I would give you some leeway. Probably if you go between negative, I don't know, like 2.5 and 2.9-ish, I'd say sure. And there we go. That is our range. Now we need to do, oh, next one. Okay. This one is not a smiley face. This is a frown. So A is negative. So I'm going to look at my chart, remind myself. It says when A is a negative, when you have this frown, y is going to be less than whatever the number is. So I'm going to write y is less than or equal to, and then I just find my number. So here we go. There's our max. Yes, that's the maximum. That's as high as it goes. And we go across. I don't know, that's negative. Yeah, negative 0.7, sure. Negative 0. I'm going to say 8. But again, because we're guessing, it's fine. So there we go. Look at that. Okay, I told students this before. Worst case scenario, you're writing an exam and it says, what is the range? If you don't know what else to write, put YER. It's right more often than it's not. It's only when you have the parabolas with either the maximums like this or the minimums like this that you have to worry about it. Otherwise, it's just what you are. Okay, we are almost done. How did it get to be this late? That's okay. Turning points. That's super easy. There is, look at that. Answer, answer, answer. Pay attention. So you can use a chart, but a turning point is really easy to see. So let's use the pictures. So here's graph number 1. Imagine you were a little bug. You're crawling down it. Crawling down, crawling down, crawling down. You get here, and now you're going back up. That's one turning point. So number of turning points, 1. Which also matches what the booklet just said. Whoops. Sorry, they're all out of order now. Number 2. All right. Brianna, how many turning points do you think it's got? How many times does that line change direction? So we're going downhill? Yes. So you change direction there, and you change direction up there. So that makes 2. So where's my file? We got a line. Cleveland, how many times does this line change direction?
Once.
Nope. This poor little ant is crawling uphill, and he's just crawling uphill. So 0. Which is reinforced by this. So you could go through and do all your constants. All your straight lines are 0. All your quadratics are 1. The only decision you have to make really is about the cubics. But I don't want to depend on my graph to do this. All right, Sammy, graph number 4. 1. Changes direction at that minimum. Excuse me. Jose, take a look at this graph. How many times does it change direction?
Once.
Perfect. Yep, you're driving in your car uphill, downhill. Cool, cool, cool. All right, number 6. Clarissa, how many times that changed direction?
Twice.
Nice. And then we got linear once. And the last one.
Linear 1.
Oh.
Linear 0.
Yes, linear 0. See, that's what happens when I try and do it quickly. Yeah, it's going downhill. Still going downhill. 0.
Twice for the left.
And twice for the last one. I should mention, I can use this up here. So these are both my cubics. What are turning points? So this one is considered to have 2 turning points. So like there's one there, and there's one there. This one, oops, turning point is 0. So you're going downhill, it kind of settles, but it doesn't really change direction. You're still going downhill. So that's what the cubic one would look like with a 0 in it. And ta-da. There's the chart. I will scan that and post it today. Probably with the practice sheet. I don't know if I have a filled in one, but this should work. I hope this makes more sense. I will do a video on end behavior. I was going to do it yesterday, but I just didn't get to it because I think it's not harder than this. It just looks scary to start with. So patience, people. What you should do today. Okay, I'm going to review biology. I'd suggest you write your biology test. But today, you have the same big piece of paper which says hand in on it. I would do this today before everything disappears. And by the way, I'll give you a hint. Oh, this one right here. This one's a constant. So number 2, put a C for constant. Your line is right there. But I would do this before you forget it. Some of you may have assignments from before to do. That's nice. But let's do this stuff while it's fresh in your mind. Please don't do page 2. If you look on the second page, there's no pictures. And Monday, I will show you how to do this part. And there's 2 options. Oh, it's really hard to see. But there's a negative squished in there. Can you guys highlight it or do something?
On the handout?
Yep. Oh, I really should retype this. But they're on.
See it.
Yeah, but not, it's hard to see. A lot of people miss it. But I would leave this. Don't do this side. Just do the other side because you know that side. And Monday, I will tell you this, it's a cheat. I'll show you how to use Desmos. You can just draw pictures of all of these and then do it. Okay, so that's it for math. And I will scan and send that out today. That's my plan.
Is there homework for this chapter? Sorry.
That's a really good question. I'm not prepared to answer that right now.
Okay.
I have no idea. Probably not right now. I'm more concerned with everybody getting caught up. That bonus assignment that I showed you guys this morning, it says end behavior, that could replace one of the homeworks if you don't feel like doing a homework, or like I said, if you've done, there's an assignment you did that either you're never going to finish it because you just hate finance with all your passion, or you hate probability, which seems to be more common. Do the end behavior one, but I would still wait because by Monday, you're going to be a pro at this. Hey, good morning, Miranda.
Good morning.
Have you been there for a while?
Yes.
Oh, I'm so sorry. Because my computer
No, it's
wasn't.
okay.
Okay.
I wondered why you weren't asking.
Oh, no, I just didn't see you. I just, my bad. Do you understand what we're doing?
A little.
Okay, well, you probably have to watch all of the video. It'd be good. So the plan now is I'm going to quickly run through the biology PowerPoint for organization, and you guys can ask any questions that you need. Oops, hello. Oh, one thing before this. If you go on to Brightspace, come on. All right, so I'm going on to biology. Okay, the computer is just not cooperating with me. I'll make it look like you. So this is biology. We got the daily videos, we got assignments, quizzes, da, 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 da. If you go into organization, and this is our last unit. This is the last unit. There is the organizational guided notes answer key. So if you have any blanks that you have not filled in, you can click on this.
Or can we fill out the whole booklet?
Yeah, truthfully, I don't care. Yep. I was trying to argue with people, but you know, there's something to be said about filling in the whole booklet and then just being able to listen
Yeah.
and not worry about stress. but it's all filled in. Not the reviews, because that's your job. That's what you get the marks for. Well, plus filling in stuff. But before you write the exam, you, at minimum, you want to make sure all of this is filled in. I'm not saying don't listen to the videos or don't watch them, because of course I talk about a lot of other stuff and try and connect it to life. But at minimum, before you write your exam, you want to make sure that stuff's done. Oh, come on. Okay, there we go. All right, slideshow. So the chapter on organization, let me get at that. Oh. Good morning, Rachel. All right. Yeah, it's part one because this one I split because it's actually pretty intensive amount of information. So the first thing we talked about was ATP. And it's lovely to understand it's adenosine triphosphate, which is this fancy molecule, but this is good enough. You've got an adenosine and 3 phosphates. And it's this last spot here where that bond will break. And if you break that, a whole bunch of energy is given off. And this is what powers almost all organisms on the earth, is this adenosine triphosphate. So everything tries to make as much ATP as they can. And yep, cyanide. I think there's other ones, but for sure what it does is it breaks this part down and you're done. So ATP, so this is the adenosine with 3 phosphates, is like a charged battery. It's got energy. And then if you break that bond, I don't know why they put the phosphate over here, but if you break the bond, then you're giving off some energy and also some water. And it just recycles back and forth. So you make this stuff in your mitochondria. Cellular respiration is the main one. It can also be made with photosynthesis, which is when the sunlight, okay, I'm not explaining to you what photosynthesis is. You know it's when the sunlight glows on the plants and it excites electrons and you go through the 2 steps as we get into detail. And then we've got the chemosynthesis, which is pretty rare, not very effective. But if you are the only organism living in an area and that's all you have for food, you're still going to be successful because no one else is there competing with you. All right, so cellular respiration occurs in the mitochondria, and that's going to be in all eukaryotes because that's what we are. So plants, fungi, animals, and protists. This is the overall reaction. I don't know if it's on the exam, but I could think a cool question is, this is 1 molecule of glucose, uses 6 molecules of oxygen, produces 6 carbon dioxide, 6 H2O, and 36 ATP. A cool question would be like, what if I had 2 of these? If I had 2 of these, you would get, well, whatever 36 times 2 is, like 72 ATP. It'll just give you multiples of that. And here's a picture of the mitochondria. They have the wobbles in them, and that just gives more room for this inner and outer membrane. Excuse me. All right, the 3 basic steps of cellular respiration. First of all is glycolysis, and it splits the glucose into 3 carbon pyruvates. You just need to know it splits it in half. We're not really worrying about what happens to the hydrogen and oxygen at this point. It breaks it in half. It requires a little energy. It produces a little energy. And anytime you see NADPH, FADH2, NADPH, NADH, any of those are just energy carriers. They carry around hydrogen, which should be easy to remember. And they also carry around electrons. So we're going to use that later. The Krebs cycle is the big one. And in it, carbon that we have is converted into carbon dioxide. And it occurs in the liquid part in the inside here. Glycolysis happens out in the cytoplasm. Okay, so in the Krebs cycle, that's when the carbon is used. I pretend this is with a C and it helps me remember that's when the CO2 is formed. We get a little bit of FADH2, again, energy carriers, and some NADH, energy carriers. But we still have very, very little amount. So it's kind of like, you know, we're having a fundraiser and for the first couple of hours, nobody's buying anything. And then we get to the last step. So now it's lunchtime and people are all showing up. We have the electron transport. Okay, and this occurs in the inner membranes. And these guys have been carrying around the hydrogen and stuff are finally being used and they make about 34 ATP. This is also where water is produced from the excess oxygen and the hydrogens are used up. But the key thing in the electron transport, it's where almost all of the energy is formed. So the opposite of that, or actually another way to make ATP, I guess, is photosynthesis. And that's going to be, if it's green, the odds are there's photosynthesis. So plants, some protists, some bacteria, and it's going to happen in their chloroplast. So remember, if you see the pictures, if it kind of looks round or ovally and it's got wobbles in the middle, so scribbly bits like this, that's going to be a mitochondria. If it's kind of the same shape, but it's got these stacks of things in them, then you know it's a chloroplast. And the simplest is carbon dioxide and water gives us glucose and oxygen. It is the reverse of cellular respiration. Okay, so here it is again. So it's still 6 CO2, 6 H2O plus light energy gives us glucose and oxygen. First step is light dependent reaction. And it's, okay, not surprisingly, requires light. And it's storing energy to use it later. So energy shines on it, shines on the thylakoids and these things. So these piles of coins, and it excites electrons and away we go. Okay. Oh, and the H2O is split and the oxygen is given off. I forgot about that part. Because for plants, they don't necessarily need it as much as we do. The second part, the light independent reaction, can occur, well, it occurs all the time. As long as there's energy, it can happen. And it occurs in the liquid part, you really can't see in here, but the background, no, there it points to it. And that's where the glucose is made. So we've got energy from before, the hydrogen from before, plus the carbon from the CO2. So 2 steps on photosynthesis. Skipping those. Skip. Chemosynthesis. Maybe 1 question on the entire test. Bank. So not likely you're going to get it, but basically it's going to be unicellular organisms, so prokaryotes, so bacteria, are going to use carbon dioxide, methane. Sulfur is a really big one as an energy source. Occurs where there's no light, such as deep ocean vents, hot springs, etc. I wouldn't worry about this, but it's just showing that carbon dioxide plus hydrogen sulfide plus oxygen can make this stuff. And then there's review part 1. Okay, extremophiles. What you need to know are these 3 words. Halophiles, which are salt-loving. Thermophiles, which are heat-loving. And methanogens. Methanogens will produce methane. So if you see philes or phylae in biology or science at all, it means loving. And they can live everywhere. So in general, what they want is a much stronger cell membrane because they're living in pretty rough circumstances. So one thing they'll do, okay, so this is what our phospholipids look like. There's my phosphate, there are my 2 lipid tails. Ours are bent. With extremophiles, they'll tend to be flat so that they can pack closer. To me, I think of it as more like plywood or something like that. Whereas this isn't going to be a very strong stack. It's going to take up extra room. Or if it's only a single layer, they'll have lots of little sticky outy bits, and it acts like Velcro to slow things down and make them more rigid. All right, and whoops. Going the wrong way, sorry. In acidic environments, you just have, okay, don't worry about that one. All right, salt. You can have charged particles on the surface that attract water and it protects it. So if the salt ions can't get to the surface of the organism, it's going to be fine. It's like I'm going to throw you in a vat of acid, but you've got a nice little wetsuit on that protects you. We have some that have pumps to get rid of salt, and other molecules, I don't know what they are in their cytoplasm to retain water. Over here is a picture of a number of proteins, but you can see they have really funky structures, and they're curled and they're all twisted. If you heat something, those are going to uncurl, tangle together, and that's not good. So what they try and do is you want to have amino acids that aren't going to do that. And what are we going to do? We're going to take our DNA and we're going to twist it up tighter, and it's going to protect it. It's going to have more GC pairs in it because it makes it stronger, because guanine and cytosine have 3 bonds and the other ones only have 2. And often they are prokaryotic and they're just simpler by nature. And they tend to be hydrophobic or water hating. So if they can repel the water, it's not going to damage them as much. And methanogens, all you need to know is they produce methane, which is CH4. So, and they live in your stomach, digestive tract. All right, now important molecules, cell membrane, cell walls, and proteins. All right, so for us, animals, we have a phospholipid bilayer. So remember these guys? The little balls are the phosphates, and the 2 arms hanging off of them, or stringing up from there, those are the lipids. Lipids is a fancy word for fats. Okay, so that's where the phospho is the phosphates. The lipids are the 2 arms, and the bilayer means there's a layer here, and it lines up with a layer on the bottom. So these lipids don't like water, so they will tend to point towards each other. And the phosphates do like water, so they will line up around the back. So this, okay, this one says this is outside the cell and inside the cell. And you can get protein channels in between. You've got proteins, you've got receptors. So when they talk about the COVID virus, there are receptors. I don't know what the shape is, but they're in your lungs and your respiratory system. And this is going to match one of the protein spikes on the outside of the COVID virus. That's why it infects it. It's not the same shape as something in your digestive system. Because if it matched that, then it would be some kind of a, I don't know, it'd be like getting the flu and, I don't know, throwing up or diarrhea or something like that. More about the same. Here's our phosphate. Oops, no, I got that wrong. No, I was right. There's your phosphate. Here are your lipids. And the phosphate, the outside, the balls are the hydrophilic. They like the water, and the tails are the hydrophobic. All right, to move through the cells. Oh, these are lovely multiple choice questions. You have basically 4. These 3, diffusion, osmosis, and passive transport, do not require any energy. Active transport surprisingly does. So diffusion, small particles, not including water, always from areas of high concentration to areas of low concentration. And one of the ways it happens is the molecules just slip between the photo. Skip back. So the molecules are so small, they can just get through in between. So normally I have students demonstrate this and they line up together and they put their arms out and they're like zombies. And then I just start throwing like little golf balls at them. And some of them make it through, some of them don't. I enjoy it. I don't throw them hard. But then I tell students to step apart. And when you have one of these protein channels, of course, stuff's going to move way faster. All right, so small particles, not including water, because, whoops, if you have osmosis, that is a special type, which is only water. And then you have the permeable membranes. Okay. I'm not even going to try and draw it. You should have the pictures. If not, Google it before you write the test. In an isotonic solution, the cells are going to stay the correct size. In a hypertonic solution, the water is going to leave the cell and it's going to get it's going to shrink down. And in a hypotonic solution, I think hypo and hippo, the cells are going to swell and they can even break. Don't worry about the fishies. In humans and animals, we have kidneys. In plants, it's the stoma on the cells that control the amount of water. All right, so if you have little particles, they can just sneak through. But if I have a big particle, then you're going to need a protein channel. If you still have more of the big particles outside compared to inside, it's just passive transport. They can just go through here, done. But you're still not using energy because there's still more on the outside than there is on the inside. All right, active transport requires energy, and it's the way to go from low concentration to high. So in this case, there's not whatever these green things are. Let's say there's sugar. Okay, so there's lots of sugar in the cell, not much outside the cell, but the cell wants it because it's our energy source. So by using some energy, it can bring these in, and that's active transport. So they're pumped across the channel. If you remember the videos, they were, I think I have a picture. Yeah, something like this would be active transport. See how it comes in and it's caught there. And then this thing changed shape and basically spits it out the other side. If it involves the cell membrane moving like an amoeba, then it is going to be some type of endocytosis or pinocytosis. So if we do this, if we have a drawing and it goes like this, and here's the food that it wants, then that is going to be endocytosis. It's going to go around. Just excuse me for a minute. Okay, double check that you guys have got all the answers. All right, back to endocytosis. So if your organism is like that, and then it ends up, man, do I hate writing with these. There you go. It gobbles it up. It's endo, because endo means inside. If it is a liquid, it's called pinocytosis, which the only thing I've got is there's like wine that's Pinot Noir and Pinot Grigio or something like that. That's a liquid. Otherwise, it's phagocytosis, which is for a solid. Also, because it is active transport, of course, it requires energy. It makes sense. You've got to, basically, it's got to surround the food particle, whatever it is. Oh, there's a good picture. So if it is spitting it out, read it this way. If you read it backwards, it is endocytosis. So exocytosis is just getting rid of it. And for some reason in biology, it doesn't matter whether it's spitting out a solid or a liquid, it's just exocytosis. All right, cell walls. All you need to know is basically in this chart, the bacteria have peptidoglycan. The archaebacteria don't. Protists, probably not going to ask you about that because there's a mix of stuff going on. Fungi have chitin, plants have chloroplasts, and we don't have cell walls, so not a problem. And they're all going to be made up of polymers, which are long repeating molecules. So if I had you all line up, I would call you monomers, because a mer is our repeating unit. And then if I had you join arms, you could make a polymer. So for plants, it's cellulose. And basically you take glucose and you put it into long chains, and that will form a starch. And if those long chains join together, you're going to get cellulose. Humans cannot digest cellulose.
What are the repeating units?
Of so glucose molecules. No. If you have fungi, it's chitin, and you can see here's the sketch or the chemical formula. It's kind of a structural one. Each one of these corners is a chemistry, and they're just showing the most important parts. It's almost identical because here, this is chitin. It's got some nitrogen and hydrogens here. This has just got an OH group of hydroxide. So it kind of shows that they're really closely related. Okay, archaebacteria, those are the most ancient organisms. And if I can remember that, maybe archae is like the beginning of the alphabet. So this is the beginning of life. They don't have the peptidoglycans. That's all you have to know. Whereas the eubacteria are slightly more advanced, and they do contain the peptidoglycans. Don't worry that it's made up of amino acids and sugars. All right, that's repeating. All right, DNA, you guys should know this down to an art. There are 4 bases, A, G, C, and T. And each one of those is connected to a phosphate and a sugar group. So I don't remember which one of the bases this one is, but there's our deoxyribose and there's our phosphate. And here's the picture you're used to seeing. We've got the beautiful double helix like this, but if you flatten it out, the bases are always attached to the sugars, and it's the phosphates that join those together. And they are held together in the middle by those weak hydrogen bonds. So a nucleotide, they show you right here, is a sugar, a phosphate, in one of those 4 bases. We know DNA carries the genetic information. Good. Okay, to make DNA, you need to break the bonds down the center, and that is something called helicase. So it breaks the bonds, and then nucleotides left over from, I don't know, like the food you ate that you've absorbed are going to come in and join. And then another enzyme called DNA polymerase basically joins them back together. So here you can see this is the helicase breaking it up at the replication fork. That's where it starts to split. And then the DNA polymerase is the one that joins them back together, because you've got these little chunks like this. And when they come in, until you get the DNA polymerase, they're not joined together, and they would just move around. And it wouldn't be those, they're held together by hydrogen bonds, so it's not very solid. Oh, sorry, wrong way. Okay, and this is just going through it one more time. 3 types of RNA. We got messenger, transfer, and ribosomal. The messenger takes the pattern out of the nucleus. That's when you, hopefully it's when you get your vaccine. That's what they're going to inject you with. So you're getting, I have a pattern for the COVID-19 spike. It's probably gone by now, but it made those proteins, and then my body was able to start a defense to it. Transfer RNA is going to move around your amino acids, and our RNA is, makes up ribosomes. So if we look at the 2 of them together, we have DNA has a double helix, RNA has got the single helix. This is going to be millions of bases long. RNA is much shorter. One thing that'll help you on multiple choice questions is if you see T's in it, nope, let me get that right. Yes, if you see T's, you know it's DNA. If there are U's, then you know it is RNA. Oh, and the sugar is different, right? Deoxyribose versus ribose. And here's your transfer RNA. So on your transfer RNA is an anticodon, which is going to match with the codon on the messenger RNA. And it's got, forget cloverleaf, it's T-shaped. If you haven't done the Bioman or protein synthesis race, you should. And rumor has it, it helps if you watch the video first. All right, there's our ribosomes. All right, protein synthesis. This is the last part. And I think you guys are lucking out because you're getting an online test, which isn't great, but you can use your notes. Don't forget, once it starts, you've got an hour to finish it. 2 parts, transcription alphabetically comes before translation. So transcription is getting our pattern. Translation is making it into the protein, making it into something new. Transcription, the DNA in the nucleus opens up, and then you get the RNA that matches. Okay, and that comes from the nucleolus. And to be careful, if this is my DNA, T is going to go with A, but A's are going to match with U's this time. So watch for that. And then once we get this piece of synthesis or RNA made, that's messenger RNA, it goes off to find a ribosome. And if you are scared by that big circle conversion thing, there are no questions on this exam about it. I wasn't sure how it was going to work or how we were going to make this work. So there aren't any. You need to know how this works, but I'm not asking, at least I don't think I asked, you know, if you see AAA, what amino acid does that count? I'll leave that for the final. Okay, so this is our messenger RNA in groups of 3, and those are my codons. There's our amino acids. So this is a better picture. This is the amino part with the nitrogen. The C with the double bond O's is the acid. And then it's just, there's different things that are bonded on depending on the molecule. And these are all amino acids. Skip, skip, skip. Step 2 is the translation. And that happens out in the ribosome. And we've got our codon here. And this is the anticodon. So A matches with U, A matches with U and G and C. And this specific anticodon is connected to lysine. So this is a transfer RNA that's bringing it in.
So on the chart, you would look up UUC, right?
Yes. And on the top of the chart, it also says messenger RNA, because for years I could never figure out which was which. And it's like, it says messenger RNA. So this is what you would look up on the chart, and it just ignores this part and it takes you straight to there. And these bonds here between the amino acids are called peptide bonds. And there's a picture of it. There we go. I didn't ask you anything about microscopes. Where's that picture go? There. That's a cool picture. I really like that one. That might even be as part of your test. But I like it because it's all labeled. So I have talked way too much and way too fast. I wanted to get this done so you had time before lunch to write your test. But let's go through this picture, just on the off chance that maybe it's on there. All right, number 1. I don't think you guys have that, but you got to stare at the screen. Driano, what is number 1? It's where all of this protein synthesis happens. It's up here. So it's this thing. You can say pass. Okay, Sammy, what is it?
That looks like a codon or something.
I'm talking about this whole thing. Let me help you. Where is protein synthesized? Starts with an R. In the ribosome. Okay, so here's my ribosome. Now, this piece here is what the ribosome is reading. Clarissa, what comes from the nucleus and is read in the ribosome?
I don't know.
Okay, Miranda, do you know? It's a type of RNA. You've got rRNA, tRNA, or mRNA. Perfect. mRNA, that's exactly what it is. I'm not sure what you're going to say, but I thought I'd just help you. Okay, so this is, we know it's RNA, see, because it's got U's in it, and it's the messenger one. These guys, number 3, hold on. Okay, yeah, let me do it right. Okay, go in order. No, I don't want to. Okay, this is how we know this is messenger RNA is a big long strand. This is tRNA because it looks like T's. There's kind of a cross thing going on. That's an easy way to tell them apart. And they also have the amino acids on there, but it looks kind of like a T, so that'd be the simplest. Now, these groups of 3, if over here, number 6, is part of our original messenger RNA, that's a codon, because that's what we start with. If it's part of the transfer RNA, then it's an anticodon. It's the matching complement. So you can see this part here. This is my messenger RNA, says GAG, and G's goes with C's, and A goes with U, and G goes with C. So then this part is the anticodon. What part did I miss? 5 down here. That is, no, brain fart. Amino acid. There it is. There's your amino acids, are these things that are being brought through. All right. That is my quick, how fast can I talk review of the chapter. This is my suggestion for today. Do whatever Brian says this afternoon. But this morning, I would do, hold on, you share. this morning we did this assignment with the practice. There is one that says hand in. It looks exactly the same or almost exactly the same. I would do that because when we went through this is the same. It's just a different set of pictures that you have to analyze. I would do that. I would, nope, I wouldn't do that. That's all I had to show you. The other thing I would do is write that test. It should be sitting in bright space for you. You've got from now until 10 o'clock tonight to finish it. So I would ask you all to write it today. Yeah, so do it, get it done, and be happy with it. It's a lot of work to reset and everything. So I appreciate if you would write it today. So even if you're not 100% confident in that material, you've got until you have to finish by 10. So if you finish you got till 8:30 to start writing it tonight or 9 o'clock. So I have faith. All right, stop the recording.